good evening, beautiful people on mediSars. I welcome you all once again. It's actually night over here. If it is your first time on this channel, please, you are warmly welcome. Okay, you're going to continue the lecture series in cell membrane structure. We know we already dealt with the introduction part of it, where we dealt with the lipid component of our foundation of our cell membrane. Tonight, we're going to make a top up of that. And please, if you haven't watched that, then please go and do so that you have a better picture of what we're going to do tonight. Okay, guys, now let's get into it. Okay, awesome people. Now, in our previous lecture, if you remember, we used some important representative structures to represent our various lipid building blocks for the foundation of the cell membrane to be what? We said that whenever that we see a polar head group with two hydrophobic tails, that is going to represent our what? Our phospholipid structures. Also, we rightly said that, and if you remember, and if you watch the videos that we recorded previously, realize that using the long-standing oval, which is shaded, is going to represent our cholesterol molecules. Awesome. Now, lastly, concerning the last type of lipids found in the cell membrane, we used, because of their similar nature to the phospholipids that we have in the cell membrane, and the only difference is that their backbones are different. One having sphingos and the other having glycerol. We consider the polar head group with two hydrophobic tails by leaving a dot in the polar head group. And we say that this is going to represent our sphingomyelin. If you remember that, fine. We are considering the sphingomyelin type because we said that it is the predominant type of sphingolipids in the cell membrane structure. Fine. Now that we have our lipid building blocks, let's begin building our cell membrane structure. Okay, now guys, let's try to look into our cell membrane structure, okay, our typical human cell membrane structure, so that we consider this to be our cell, okay? This is going to be our cell with a cell membrane and a central nucleus, right? Awesome. So guys, what we're going to do right now is that we're going to open up our cell membrane structure so that we can fit in these macromolecules or these building blocks that we've established so far, okay, inside our cell membrane. Fine, but before we do that, there is this concept that we go to respect, and what is that? Looking at our cell that we have on the board right now, it has two important environments, okay? The environment within it, confined by its cell membrane borders, or which we call it the intracellular environment. Take note of that. Intracellular what? Environment or intracellular space. And then it has this environment exterior to it, surrounding it. Okay, so because of that, we say that this environment is termed as what? The extracellular environment or extracellular what? Space. Take note of this. Now, why do you have to know this? With regards to our extracellular environment or space, okay, later we realize that there are some important macromolecules like glucose, proteins, some lipids, and some electrolytes like calcium, magnesium, like some sodium ions and the potassium ions. And then these entities are all floating in water, okay, inside the extracellular space. These entities that we're talking about right now, they are all floating in water. So because of that, we're saying that our extracellular space is a water-filled space. It's filled with water. Okay, it's a water-filled space. And in more scientific terms, you say that it is an aqueous medium. Take note of that. It is what? An aqueous medium also. Now, why do you have to know this? We're going to know that very soon, okay? Let's come back to our cell membrane structure. Fine. So because we have our phospholipids to be the predominant type of lipids in the cell membrane structure, we're going to consider it first. So let's open it up right now and see how the phospholipid structures are neatly arranged in our cell membrane, okay? Awesome. Fine. So how are our phospholipids being arranged in the cell membrane? Okay. This is how we answer this question. The phospholipid structures, okay, are arranged in a linear chain fashion, in a way that one is being placed by each other side, as I'm illustrating in the board right now, to form this linear configuration, filling up the entire space of the cell membrane. They are arranged in a linear chain like this, okay? And remember, in our previous lecture, we said that the phospholipid structures have two important chemical sections, okay? Fine, we said that the polar head groups are the hydrophilic section, meaning what? We said hydrophilic means what? It is the water-loving section of the phospholipid structures. Remember, water-loving section or domains of the phospholipid structures. And then you also realize that these tails that we have beneath the heads, okay, are the water-hating. Please take note of this. Very, very, very important to consider. Or the hydrophobic domains of our phospholipid structures. Fine. Now, knowing this, we come back to a question: how are these phospholipid structures being configured or oriented in our cell membrane space? How are they being arranged? Fine. So now, respecting our water-filled extracellular space, okay, and knowing that our phospholipid structures have two distinct portions, water-loving and water-hating portion, do you think that in bringing this one over here to fill this space, the water-hating domains are to be directed towards the water-filled space, or we should hide them away from the water-filled space by directing instead the water-loving domains of our phospholipid structures towards the water-filled space? Yes, exactly. We said that. What we're going to do right now is that we're going to make sure that the hydrophilic domains, which are the polar heads, are the ones directed towards the water-filled space, because they love water and they like to have that kind of relationship with water. So they will be facing this water-filled extracellular environment so that we can hide away the hydrophobic tails between them. You see how the cell membrane phospholipid structures are being configured? Wonderfully arranged over here. Fine. Now, because you have other lipid building blocks forming up the cell membrane, let's try to add them over here. So our cholesterol molecules are somewhere intercalating between the hydrophobic tails of our phospholipid structures over here. Okay? Yes. This is how our cholesterol molecules are being arranged and positioned in cell membrane structure. Take note of that. So let's try to add some more sphingomyelin molecules over here. So we have some 3 sphingomyelin molecules because they are just few, forming about 4% in the cell membrane structure, if you remember from the beginning of the lecture. And then we have, because the phospholipid structures are forming a predominant type of lipids in the cell membrane, let's just close the gap by adding some more phospholipid structures. Okay, awesome. So guys, this is how our cell membrane structure is being arranged. This is how our lipid building blocks in the cell membrane structure are being arranged in the cell membrane. Fine. So let's try to intercalate between the hydrophobic tails of these phospholipid structures with some cholesterol molecules in here. Yeah, it's very, very important to take into consideration. Fine. Now, the question we're going to ask ourselves right now is that in a typical human cell membrane, is this how it looks like? Is this how it typically looks like? And the answer is a big no. Why no? Because this intracellular space that we have over here also happens to be a water-filled space. Why is it then contributing to the reason why this is not the entire nature of the cell membrane? Because remember that we make sure that these hydrophobic tails are directed away from the extracellular space, okay, so that they can be hidden from this unfriendly extracellular space by showing to the extracellular space, water-filled space, the hydrophilic or the polar head groups of the phospholipid, the sphingomyelin structures. But here is the case whereby, unfortunately, the intracellular environment too is a water-filled space. It is also an aqueous medium. Isn't it an unfortunate situation to encounter? Yes, because it is also an aqueous medium. But these hydrophobic tails are water-hating. So how do they manage to be in this unfriendly environment? So it means that this shouldn't be the typical nature of our cell membrane structure. There should be something added up to it. And what is that thing? Okay, so that thing that's going to be added right now is what is going to solve the problem at hand. And what is that problem that we have at hand? The problem is that, do you remember that we decided to face the hydrophilic heads of these phospholipid structures and then the sphingomyelin, the cholesterol molecules that we have in this chain towards the extracellular environment, which is water-filled, in order to hide away the hydrophobic domains of these structures towards the intracellular environment? And now later on, we've come to understand that the intracellular environment is also an aqueous medium. It's also a water-filled space just like the outside. And now we are facing the hydrophobic tails towards it. That is a problem, right? So how do we solve this problem? In solving this problem, that is where we add up another chain of phospholipid structures down here to cover up these hydrophobic tails. That is how we solve it. Fine. So how do we do it? We know that we have these chains to be linearly arranged. These phospholipid structures, they are linearly arranged in this fashion. Fine. So in solving this problem, do you think that we are going to make sure the hairs are pointing up like this with their tails pointing down to close up the hydrophobic tails of the outer chain? Do you think that's going to help? No. Why? Because even though if this is how it's going to continue to close up the gap, yes, the hydrophobic tails are going to be covered all right, but the lower chain that we create over here will still have their water hitting portions facing the inside aqueous medium as well. So that's not going to be a way of solving the problem. So the only way of solving the problem is what? Turning the lower chain phospholipid structures upside down, whereby they will be having their head groups facing down like this and their tails pointing inside. So guys, if it is how it's going to continue like this to fill up the space as I'm indicating in the board, don't you think that we are going to safely hide away the outer chains hydrophobic tails from the intracellular environment? And also by so doing, you're going to hide away the hydrophobic tails of even the inner chain from the intracellular environment. Fine, that is how the cell membrane structure is built. That's how it's built. So we have the inner chain having their phospholipids head groups facing down in order to hide away the hydrophobic tails into the interior of the cell membrane structure. And then that is helping us to cover up the hydrophobic tails of our outer chain as well. Take note of that. Fine, so this is how we build our cell membrane structure. So let's try to add some cholesterol molecule to be intercalating between these hydrophobic tails that we have in the lower chain structures. Fine, so let's try to close up our cell membrane safely. Awesome. So guys, this is how nature wonderfully builds the cell membrane structure. So as you can see, the cell membrane is a double layer. It has two layers. So because of that, that is where we come to this concept. Why do we say the cell membrane is a phospholipid bilayer? Because as you can see on the board right now, it is into two layers. And you know that the predominant type of lipids in this is the phospholipids. So for that matter, we see that the cell membrane is also termed as a phospholipid bilayer. So what I'm saying right now, the reason why we say the cell membrane structure is a phospholipid bilayer is that it is into two layers, as you can see on the board, and then the predominant type of lipids in the cell membrane structure is what? It's phospholipids. That is why we say that the cell membrane is a phospholipid bilayer. Fine. Now, last concept. Looking at our cell membrane structure, yes, we know that it is into two layers. You have the outer chain and the inner chain. Now, is that how in cell biology we call them? Do you call it outer chain and inner chain? No. Actually, this is how you term them. Because this outer chain is facing the outside or the outer part of the cell, we term it as the outer leaflet. Okay? Outer leaflet or the exterior leaflet. Take note of that. And then, because the inner chain is facing the inside of the cell, okay, we also term it as the inner leaflet. But there is another name. Because this same inner chain happens to face the cytoplasm of the cell, we can also call it the cytoplasmic leaflet of the cell membrane. Take note of this. You have to take note of them so that you don't get lost in some of the rules that I'm doing, because they'll be using that along the outer leaflets and the inner leaflets. This is what they be. The inner chain is referred to as what? The inner or the cytoplasmic leaflet, whereby the outer chain is what we call the outer leaflet or the exterior leaflet. Okay. Now, I want you to take a very close look at these two double layers of the cell membrane structure. Just take a very critical look at the outer leaflet and compare it to our inner leaflet that we have on the board right now. Guys, can you point out some differences between these two layers? Look at it critically for me. Don't you see that in the outer leaflet there are some sphingomyelins in there, but in the inner leaflet there are none? Yes. In fact, is it happening by chance in our illustration, or that is what is happening in a typical human cell membrane? I must tell you that that is what is happening in a typical human cell membrane. So in a typical human cell membrane, entering into the outer leaflet, you find sphingomyelins amongst the cholesterol and the phospholipids in there. But in the inner leaflet, there is none. Take close note of this one. So what I'm trying to say right now is saying that in the outer leaflet and the inner leaflet, there are some differences that can be pointed out. And one difference that we now know is that the sphingomyelin compositions are different. whereby in the outer leaflet there are sphingomyelins, and in the typical human cell membrane inner leaflet there are none. So this is the reason why, in fact, it's contributing to the reason why we say that the two layers of the cell membrane structure are asymmetrical. What are you trying to say right now? You are saying that the outer leaflet and the inner leaflet layers of the cell membrane structure are asymmetrical. And now we know that one reason is because of their difference in the sphingomyelin compositions. There are some sphingomyelins in the outer leaflet, but in the inner leaflet there are no. So let's try to put it on board right now. So we are trying to come out with some causes of the asymmetrical nature of our cell membrane. Causes of the asymmetrical, very, very important that you have to take note of. Causes of the asymmetrical or unidentical nature of our cell membrane. Now we know that sphingomyelin composition differences is one of them. Sphingomyelin compositions. Take note of that. There are other reasons why we are saying that these two distinct layers or leaflets of the cell membrane structure are asymmetrical. Now, take note of the first one being the sphingomyelin composition differences. What are the other reasons? Again, let's come back to our cell membrane structure, our typical open up cell membrane structure. Take another close view at it. Okay, compare the outer leaflet with the inner leaflet. Can you count the number of cholesterol molecules that you can see? Just count the number of cholesterol in the outer leaflet and compare to the cholesterol molecules that can be found in the inner leaflet. Are they the same? No. It looks like there are more cholesterol in the outer leaflet as compared to the cholesterol molecules found in the inner leaflet. I didn't tell you that this is not happening by chance. In a typical human cell membrane, cholesterol molecules are more concentrated in the outer leaflet as compared to the inner leaflet. So it's also contributing to the reason why we are saying that our cell membrane structure is asymmetrical. So let's try to add up. So now I'm going to talk about right now, we're saying that the cholesterol compositions, okay, differences is also contributing to the unidentical nature of our cell membrane. Fine. Now the question is, why is it that there are more cholesterol in the outer leaflet, but there are less cholesterol in the inner leaflet? Why? What is causing these differences in cholesterol concentrations in the outer leaflet and the inner leaflet? And the reason is very simple. It is because cholesterol molecules have more affinity for sphingomyelin than phospholipids. Remember, in the inner leaflet, you have only phospholipid structures plus some cholesterol in there without sphingomyelin. But in the outer leaflet, you find some phospholipids alright, but together with cholesterol and sphingomyelin. And now we are saying that cholesterol molecules have more affinity for sphingomyelin than phospholipids. So you can tell me now that this is the reason why cholesterol molecules are more concentrated in the outer leaflet than in the inner leaflet, because in the outer leaflet there are sphingomyelins, but in the inner leaflet there are no. Cholesterol molecules have more affinity for sphingomyelin than phospholipids. It's very simple. That is why they are trying to be concentrated in the outer leaflet than the inner leaflet. Please take note of that. So now, what have we done so far concerning our asymmetrical nature of the cell membrane? We now have two reasons contributing to the reason why these two layers of our cell membrane structure are asymmetrical. One being our sphingomyelin composition difference, and now we know that the cholesterol compositions are also changing in these two distinct layers of the cell membrane. There are other more reasons. Later on, you remember we said that in previous lecture we said that the cell membrane structure has some carbohydrate molecules, okay? Some carbohydrates taking some places in the cell membrane structure. You realize that there are just some traces taking about 3% out of the raw chunk of the cell membrane. Yes, I must tell you now that all significant cholesterol, in fact, cholesterol that can be detected are peculiar to the outer leaflet. They are found in the outer, associated with the phospholipid structures and the sphingomyelins found in the outer leaflet. But when you come in the inner leaflet, you won't see any recognizable or significant type of carbohydrate in there. So what are you trying to say right now? Cholesterol carbohydrate molecules are associated with the outer leaflet, but not significantly in the inner leaflet. So the next reason why we are saying that our cell membrane structure is asymmetrical is what? Due to carbohydrates, what? Composition differences. Take note of that one also. Also, there is some more differences to also add up. Now, later on we talk about the protein composition of our cell membrane. Realize that some proteins are found within these phospholipids and cholesterol makeup of our cell membrane. Some of the proteins are just placed in the cytoplasmic phase of our cell membrane structure. Some of the proteins are just placed on top of the outer leaflets. We realize all that in later lectures. Now, what we need to know about proteins now is that proteins found in the outer leaflets are a little bit different from the proteins found in the inner leaflets. And it's also applying to the reason why these two leaflets are tending to be unidentical or asymmetrical. So there are what? There are different proteins associated with what? So due to what? Protein composition differences, okay? So you find some different types of proteins found in the outer leaflet, but when you get into the inner leaflet, you realize that there are some different proteins that you haven't seen in the outer leaflet before, associated with what? The phospholipid structures and all the lipid molecules in the inner leaflet of our cell membrane. Awesome. Now, one last important difference that you have to take note between the outer leaflet and the inner leaflet, contributing also to the asymmetrical nature of the cell membrane, is about the phospholipid types. Remember, we said that later on we talk about the types of phospholipids that we have in the cell membrane. This is the time to talk about that. Fine. It is looking as if that in our illustration, although because we are using black marker to represent all our phospholipid structures, it is looking as if that the phospholipid types found in the inner leaflet are the ones found in the outer leaflet. I must tell you that, in fact, there is a big no. There are different types of phospholipids in the cell membrane. For that matter, the types of phospholipids found in the inner leaflet are different from the types that you can see in the outer leaflet. Take note of that. So now, what we're going to do right now is, what are these types of phospholipids that we have? What are these types of phospholipids that we have? And therefore, which of them are peculiar to the inner leaflet and which of them can be found in the outer leaflet there? Fine. So that's what we're going to do right now. So you're going to permit me to clean this part of my book. Okay guys, now so to understand the different types of phospholipids that we have, okay, you've got to visit back to the typical nature of a phospholipid, okay, what we've dealt with in the previous lecture, and try to visit it back so that we can get a clear understanding of the different types of phospholipids that we have and what is really making the difference amongst them. So let's try to go back to our typical nature of phospholipid. Remember we said the phospholipid is made up of what? A backbone called glycerol, which is a 3-carbon compound with 3 hydroxide ions and then some 5 protons binding to them. Awesome. And we said that binding to the carbon 1 and carbon 2, okay, the hydroxide ions of carbon 1 and carbon 2 are what? Fatty acid chains, yes, making up the diacyl glycerol, if you remember all that. And then binding to our carbon 3 over here, the hydroxide ion of our carbon 3 over here is what? A modified phosphate ion. And that is where we said that modified phosphate ion is simply a phosphate group or a phosphate ion which is having an extra molecule binding to it. Fine. So now it's very simple to know the different types of phospholipids that we have. It's very simple. Okay, so to talk about the type of phospholipid is simply what is actually binding to the phosphate ion, causing it to be modified. Okay, so the type of phospholipids that we have is just simply the molecule that is binding or that is modifying the phosphate ion, which is binding to the hydroxide ion of our carbon 3. As simple as that. Fine. So now let's get into it. So in a particular type of phospholipid, the molecule that is binding over here is a very important structure in physiology called choline. We call it choline. Okay, choline. This molecule, when it binds to the phosphate ion to render it modified in the entire structure of phospholipid, we call this type of phospholipid phosphatidylcholine. It's so strange in the beginning, but later on we realize that, hey, it's very, very simple. We call this type of phospholipid phosphatidylcholine because that molecule that is rendering our phosphate ion modified is choline. It's so simple. Now, take note of the phosphatidylcholine type of phospholipid. There are other modes discovered. in a different type of phospholipid, what is happening is that it is not a choline molecule that is binding to it, but actually a molecule called inositol. Okay, fine. So now, because now we are having inositol but not choline, we call this type of phospholipid simply phosphatidyl. It's so simple. Phosphatidyl inositol. So you realize that in all the different types of phospholipids that we have, they will be maintaining the phosphatidyl prefix. What will be changing is that extra molecule rendering our phosphate ion modified. That is what is changing. And you all have this prefix phosphatidyl. So the rest of them, you just tell me, okay, as soon as I change this one, you just simply tell me the name of the phospholipid. So now, in the different type of phospholipid that we have, okay, there is neither choline nor inositol, but a molecule that we term it as ethanolamine. So now you guys can tell me so simply that in this type of phospholipid we will term it as what? It is simply going to be our phosphatidyl ethanolamine. So easy, right? Fine. Now, in another type of phospholipid, I think that is concluding the important phospholipids that we have in the cell membrane structure. You will never find the choline over there. Neither will you find anything like ethanolamine. You will find anything like inositol, but a molecule called serine. Serine is a type of amino acid. Okay, so when a serine molecule binds to the phosphate ion of this cumbersome structure that we have on the board representing phospholipid, we term this type of phospholipid what? Simply phosphatidyl serine. So simple, just like that. So guys, these are the 4 main types of phospholipids that can be found in the cell membrane structure. But remember we said that in the types of phospholipids, because sphingomyelin type of sphingolipids also happens to have some phosphate ion in its structure, you said later in previous lecture that it is mostly included in the types of phospholipids that we have. So in some literatures, you realize that they will mention 5 types of phospholipids, whereby you consider choline, phosphatidyl inositol, ethanolamine, serine, and sphingomyelin. Okay, but now you know that sphingomyelin is actually a type of sphingolipids, but it's mostly included in the types of phospholipids because of its bearing a phosphate ion. It's as simple as that. Okay, so take note of that. So guys, these are the 4 main types of phospholipids that can be found in the cell membrane structure. So then we come back to the question, which of them can be found in the outer leaflet and which of the 4 are found in the inner leaflet? And the answer is very simple. And what is that? Now, in the outer leaflet, it is just the phosphatidyl choline that can be found in there. It is just the phosphatidyl choline. So in the outer leaflet, if you are trying to place O over here to represent outer, it's just phosphatidyl choline that can be found in the outer leaflet of our cell membrane structure. The 3 are all predominant in the inner leaflet of our cell membrane. It's so simple as that. And guys, this again is applying to the reason why our cell membrane double layers are asymmetrical. So let's try to add it up over here. So finally 5, we are saying that the different types, the types of what? Phospholipids, okay, found in the outer leaflet are different from the ones found in there. Now we know that it's only the phosphatidyl choline that is predominant in the outer leaflet, but the phosphatidyl inositol, ethanolamine, and serine are the predominant type of phospholipids that can be found in the inner leaflet of the cell membrane. So guys, basically we have 5 important reasons why we are saying that the outer leaflets and the inner leaflets of our cell membrane structure are asymmetrical. Okay, so we've said what? The single amino compositions are different, cholesterol contents are different in both leaflets, and then carbohydrate too different, and protein composition too vary. And now finally we know that the types of phospholipids found in these distinct layers of our cell membrane structure also vary. It's as simple as that. Fine. So guys, it is very important to know these things because in some way somehow appear in some of the exams that we write. Okay, so take note of them.